Boris, you were talking about your experiences with Crick, and I know that you told us, but say again what his reaction was when he heard or read Watson's book.
Oh, yes, this I didn't hear from firsthand, but he was so upset at Watson's book that he didn't speak to Watson for many years because Watson exposed their things that were very personal, and Crick wasn't happy at all. But I guess finally they made up and forgot the issue there.
And also about what the name of the book was originally.
The name was Honest Jim. And the reason for that name is because Watson wrote it as it was, and was a shock to the community of scientists, because these were things that scientists shouldn't say outside, were certain, like a religious organization. You don't go around telling people that you have a good party with wine and things like that. So these were inner things that, intimate things that were not appreciated much. Many scientists were mad at Watson at the time. Then, I guess, biography became more common.
Are there any stories or anecdotes that you would like to share? Mark, do you remember any?
How about for Ted Puck?
Nothing
He was a famous guy.
Yeah. But I didn't see him. He wasn't in the lab very much. So I didn't have much opportunity to interact with him, which was unfortunate because I would have learned a lot more about cell biology. So I didn't spend much time with him.
How about Boris' experience with Salvador Luria and some of the Fage group? They were sort of a very close circle, weren't they?
Which group?
The Fage group.
Oh, yes. Ted Puck was a member of that club. But the Luria anecdotes could fill up a book. There were so many. The one that just flashed was in Illinois when we were graduate students. We were very fond of practical jokes, and someone rigged the tape, so we were at a party. Those days there were lots of parties. It was hard work, but hard partying too. And someone rigged the tape that was played on the radio. We were in the middle of the party, and the announcer said, latest news, Professor Salvador Luria from the University of Illinois has been accused of seducing one of his assistants. And Salva's reaction, he was drinking. All of a sudden he heard it, and he paid attention, and then he said, I didn't do it. That was one of the things that, but Luria had thousands of anecdotes. He died about 8 months ago, I think. So many stories that it's almost impossible to
Are there any, as we finish this segment, any last thoughts that either or all of you might have about the directions that you see the sort of the frontiers of biology entering, and what are going to be the interesting and crucial questions in the near future? I think we've talked about human genetics quite a bit. That's obviously going to be one. But what about some other areas? It could be both in either research or technology, or where you see that a technology or instrumentation, again, aside from the human genome, which we touched on, that will be important, that should be developed or might be developed.
What are you looking at?
I'll start with that.
Yeah, I've got something to throw into this. There's a new technology emerging called confocal imaging microscopy, and confocal meaning the ability to focus down into a tissue or a cell and view only that focal plane and not be influenced by what's above or below. So you see a nice clean image as if you had mechanically sliced through an object. What makes that especially interesting now is that there are some emerging, developing dyes that are specific for metabolites or ions. For example, there is a dye called Indo-1, which is specific for calcium. And when this Indo dye binds to calcium, it changes the emission spectrum of the fluorescent Indo molecule. Indo calcium has a different fluorescence than Indo without calcium. Okay, so with that technology, you can now look down inside of a living cell. You can stimulate that cell with a compound or light or whatever other external influence and watch how that cell responds in terms of opening its cell membrane to the passage of calcium. So you can begin to look into the physiology of what's going on inside a cell. A project that we had started at UCSF just before I left, we were taking a pancreatic islet, which is an islet from the pancreas, and the islet contains 4 cell types: alpha, beta, delta, and PP cells. These cells work together in an islet as a small micro tissue, as a community of cells, and they interact in the regulation of the production of insulin in response to glucose. So this little islet, which can be on the order of 200 microns in diameter and a cluster of 3,000 or 4,000 cells. This islet is a community of cells that interact, and you can observe this interaction by using a dye like this Indo-1, looking down inside the cell. You can then challenge the islet with a change of glucose level, and then watch how calcium acts as a messenger of communication between these cells. So my point is that this kind of a technology will now allow you to watch the interactions among cells in a tissue. For example, one thing that I would like to have done, wasn't able to, is to look at a small tumor in situ, that is, in a piece of tissue, and then study what happens at the interface of that tumor and the surrounding normal tissue. How does this tumor interact with the surrounding tissue? Is there some detectable chemical process that's part of the penetration of the surrounding tissue by the tumor as it grows? I think that these kinds of tissue-related questions are going to become important. So instead of talking about single cells, which is essentially all we've talked about, now we should begin to talk about combinations of cells, communities of cells. And that's a much higher perspective and will be very important in seeing how cell communities interact. So I think that's an emerging area.
What do you do, Martin? Where do you see some of the exciting possibilities?
Well, I've been fantasizing about where biology is going in the next decade or two for a few years now, just on and off for the fun of it. And on the basis of the notion that the revolution in biology in the last half of this century was based on the discovery of the DNA structure and how DNA works. And that kicked off the field of molecular genetics, which has blossomed incredibly in the last 30 years or so. And in principle, in some sense, that field is all wrapped up in the sense that much of the general principles are known, and a huge amount of work remains to be done in working out all sorts of important details and important implications of all of this. But to a large extent, you could say, in some sense, the field is wrapped up. Where is the next big biological breakthrough? And I think it's already started, and that is the brain, how the brain works, and the connection between, and therefore how the mind works. And I mean, here's the most important organ in the body by far, the most complex, the least understood. And so my guess is, and I don't know if that guess is worth much, but my guess is that neuroscience is the big thing in biology and medicine in the next 10 or 20 years. And if I were jumping in from the beginning, I would jump into that.
I think you share some of that something, don't you, Ryan?
It's interesting that I share Max's view of what's next here, and I am into it right now because we are doing microorgan cultures, which are to be little organs that Max described, except that we isolate them from tumors rather than from the pancreas. And I share with Marv about what is in the future, except that I wouldn't jump into neuroscience, mainly because what I was telling you yesterday, this analogy of muddy fields and semi-solid fields, I think neuroscience is not ready yet to jump in that. I see, maybe 10, 15 years of it. So if I were 25, I would jump. But I would jump at 60 on it. With regard to what I think is a phenomenon that fascinates me most is the long-range effects in animals and humans. The fact, for instance, that the person reaches puberty with such accurate timing, like accurate, I mean between 12 and 14, let's say. Most of the people reach puberty. And if you try to think a mechanism to do that, it's impossible. We don't have a slightest idea of how to do that. With his brain, at least, we can make models with computers or things. There are hundreds of models of how the brain works, but no one I know has a good model of how an interaction of 14 years can lead to such precise effect. That's what fascinates me. But I will jump into that because I have no clue as to how to solve it. But it's fascinating.
It's clearly impossible. This can't happen.
Right.
Well, I think this has been fascinating for all of us, and thank you very much for sharing your ideas and thoughts with us. Thank you.
Oh, sure. It's been fun.
Now you have to stay here.
And Brian's going to do his number on you.
What's Brian's number?
You'll see. You'll see.